Welcome to the class. This class we shall be treating modifier genes. You know, there are quite a number of genes that may be found in any one particular animal or species. Okay, but oftentimes these genes do not necessarily act independent of each other. They co-interact with themselves. So in this case, we have some genes that their form of interaction is not necessarily to mask another gene, in other words, to prevent another gene from any form of expression. But however, what modifier genes do rather is to determine the extent to which another gene is expressed. So they allow another gene to be expressed to the extent to which the modifier gene permits. I think it would be better to look at a practical example, hypothetical example. Let us say we have a locus B. The allele B is a code for coat color in mice. All right. So let us say if the allele B is present, that mouse is going to be black. But if it's the recessive form of the allele, let us say the mice is going to be brown, or it codes for brown. All right. So in other words, if there is dominance, dominance is intra-locus interaction, that is interaction between genes of alternate forms of genes on the same locus. So these two alleles here, they are on the same locus. If there's dominance, if this is B and you have a genotype like B, small letter b, you know that this animal will be phenotypically black. But in this case, we are talking of a different gene altogether, not this very B, a different gene on a different locus acting, and not to mask this, but to determine the extent to which this gene is going to express. So let us say that we have a different gene on a different locus that goes by the allele small d. Okay, B, a capital D and small d. In other words, if there is capital D here and it has dominance over there, if there's capital D here, there's gonna be full, okay, full expression, okay, or full intensity of expression. And whereas if this one is small d, then there will be low or something like low or no expression, okay, or no expression at all, or low. All right. So in other words, a modifier gene, let's just say D is the one we're considering a modifier gene. Either color that the mouse has, whether it is a capital B or small b, it is this gene, this modifier gene, that will determine whether the color will be expressed with full intensity or with low intensity. So if this location, this locus here determines the color, all right, but this location here determines the expressivity. So now if a mouse is black, the extent to which the mouse is black will be determined by the presence of capital D or small d. So if the mouse is black, but here there is a capital D and it dominates, it means that mouse will be intense black. All right. So, but if the mouse is black and then here there is just maybe small d, in that case recessive, it then means that the mouse will be black, all right, but will be light black. Similarly, if the mouse is brown, that indicates that there is no capital B here, let us say homozygote recessive, small b and small b. That means in that case, the mouse will be brown. But the extent of the brownness, the thickness of the brownness, the intensity of the brownness will be determined by this other gene, this modifier gene that is on another locus. And what is happening there? If it is also small d, it means that the mouse that was brown here will be light color in brown, in being brown. All right. We can go further and just look at, let us say there is a cross between heterozygote, a mouse that is heterozygote for this trait and heterozygote at this locus, heterozygote at this locus, heterozygote at this locus. So this place will be, this is the way that such a mouse will be. So if like there is a cross between this and another mouse of the same heterozygosity, like this, for example. Okay, so you're going to see some interesting array of phenotypes. So we may want to look at the gametes that will be produced here. The gametes that will be donated here for such a cross will be B, capital B, and and the B, small b, and then here will be B, capital D, and then this will be small b and small d. All right. So if we have a Punnett square, okay, a Punnett square, we distribute this gametes in the Punnett square. We're gonna have BD, B small, and then small b, the D, and then small b, small d. All right. So let's say over here too, that's for the one of the parents. These are all the gametes. One of the parents will donate. The other parents will donate his own gamete this way. The same thing because they are both heterozygotes. Okay. So here will be small d, and here will be B and D, and here will be small b and small d. So you see that mating, we can also complete the square like this. Okay, so we have a line here, have one here, have one here, have one here, and this way, and this way. Okay, so these are the possible offsprings that we're going to have. This and this will give us radio pattern. This will give us B, B, D, D. Hope we're together. Okay, sorry. Then this will be B, B, D, small d. And then this will be B, small b, and D, small d. And then the other one will be capital B, small b, and capital D, small d. All right. So let me just stop there for want of time. But you can see that right away from here, you can see that if this is coding for black, it means the phenotype of this first mouse will be black, intense black. See this? Full expression. Intense black. So this mouse here is going to be intense black in color. Let's come check this one. It will be black and also intense black because there's dominance here. All right. It's dominant over. There's something here. Black, there's dominance, intense black. Black, also intense black. Okay. We can go on and see other variants. Here we can go have black, and okay, also intense black. Then we have here also black in color, but you see light black. You can see because there's occurrence of small d here. So that means that will be light. The blackness will not be very intense. Okay, so you can go on like that. If you come here, you complete this, you won't have something like this. In fact, even if you go this way, you're gonna have a sorry, okay, you're gonna have this. There's also dominance, but you can just complete the rest. But by so doing, you will find out that some of the animals that we have a certain color, but the expression of the color, the intensity of the color is now determined by what is happening on this locus. So that's just what modifier genes are. If you like the lecture, I will ask you again and again, subscribe to the channel like and share the video with your friends. All right, see you in my subsequent videos.